Hello everyone, welcome to STEMming, and today we're joined by Estelle Nassar from the Francis Crick Institute. So Estelle, for those who don't know you, could you please introduce yourself?
I am a research scientist at a neuroscience lab at the Francis Crick Institute, which is one of the biggest biomedical research institutes in Europe, let's say. What I do is I'm not essentially in a lab, first of all. You have kind of many different roles. Of course, you have the PI who's in charge of it, and then you can have PhD students and you can have postdoctorates, fellows. Essentially, it's anyone who's already done a PhD and who wants to stay in research would become a postdoc and then later on become a PI. But you can also have sort of people who haven't done a PhD, as because I haven't. I've kind of recently graduated with a master's, and I wanted to try out research. So this role, this lab research scientist role, is kind of a role that many labs have, where you can kind of bring your specific skills into the lab and help out through different ways. And I kind of am mainly involved in two different projects.
So when did you first become interested in STEM?
That's a good question. I think I was mainly just good at science in school, and I just didn't like everything else, so I just stayed in science, and I just had a passion for it from the start, I guess. I think I had quite a few good teachers in chemistry, science, in chemistry and biology and physics even. And yeah, they I think they helped me out a lot in terms of driving my passion for STEM in general. Yeah, I would say that.
Do you think you maybe had like one moment where you're like, oh, like kind of click, like I want to do science? Like maybe you had to choose between different, or maybe two paths or three paths. Was there any maybe
To
memorable
be
one?
honest, I don't I don't know if I had that to be honest, I think I had that moment. Well, I mean, I didn't really study science at university. I studied engineering. But when I was going to my uni's open day, there were essentially, yeah, drop-ins for different engineering fields. And I did not even think about like applying for biomedical engineering, which is what I studied in the end. But when I went to the drop-in and they explained what it was, I was like, okay, this is really cool. This is it for me. I think it was that. I think that was a moment that things were like, yeah, I think I'm going in the right track. This is a really cool field. And essentially, the good thing about biomedical engineering is that you still have it is an engineering degree, but you still focus a lot on the science as well, because of course the field is all about applying engineering principles to the medical field, to I guess healthcare in general. So you really have to stay on track with the latest scientific findings and stuff like that. So I think it was a good mix of biology and also I was really good at maths as well. So it was a good mix of everything that I liked. So maybe that was kind of like the moment where I was like, yeah, this is the path for me, I guess. Yeah.
So just to clarify, you like applied to do biomedical engineering straight
Yeah.
from the off like from Newcastle. Okay,
Yes.
cool.
Yeah. That was my, and as I said, I didn't even have that on my radar before going to that open day, that one open day. So yeah, I think that kind of changed my path, and I was like, yeah, and I ended up loving it. So I think I think I would say open days are really really important because of that. But I don't know how that happens nowadays, to be honest. But hey, that's another story for another day. But
I
yeah.
mean, yeah, for one of my open days that I went to, they demonstrated a physics principle that just got me really interested and kind of sparked what I talked about in my personal statement. So yeah, no, I completely agree with you about the whole open
Yeah.
days thing.
Yeah.
So what was your goal growing up, and did that change as you grew up, or as you went to more open days or had experiences as you grew up?
So I wouldn't say I had a clear goal. I mean, my goal changed quite a few times. As everyone interested in STEM, probably like at some point would have been interested in medicine, right? So I was like, should I do medicine? I don't know. I did consider it, and then I never really knew exactly where I was going to end up, but I knew 100% it was going to be in STEM, and that hasn't changed. Having worked in a science for that's close to a year and a half now. I can't really see myself any other in any other sort of field. Science and engineering in general, just the innovation in those fields is kind of what I want to stay, I want to be involved in. And yeah, and I don't, yeah, I don't know how people have kind of this clear path from the start. I didn't, but I'm figuring it out as I go, you know? That's kind of, that's life as well.
Yeah. So where you work now, what is a typical day like in your life,
Yeah.
regarding like your job, pre-COVID and then
Okay.
post-COVID?
Pre and post COVID. Oh, okay. And during COVID, I guess. So, well, pre COVID essentially is I mainly am a computational person in the lab. So it wouldn't be close to, I would stay in my, I would stay like at my desk most of the time, but I do help out a lot in the lab with different things. So let's say, you know, I would stay at my desk for like a couple hours in the morning, but then I can go help out one of the postdocs with their behavioral experiments. And just to clarify, with behavioral experiments, we mean we work on mice. So that's like a first thing to clarify. And when we also kind of are interested in behavior in mice, so we do a lot of sort of, we essentially let mice do their thing and kind of observe them, you know. That's, so it's not there's not that much skill required, so I can kind of help out with that. So I could be helping out with that, and I could also, let's say in the afternoon, I could maybe be helping one of the PhD students with, let's say they're coding up something, because I do have, I would say a bit more experience with coding than they do. So if they have any problems, I could help them out. And, you know, every time, you know, we all have lunch together, usually at this canteen that we have. So yeah, and that's, and we always leave time for a chat or two, you know, during the day. The environment of a lab is very nice. I've loved that from the start. That's kind of a general day for me. I mean, of course, with COVID's labs, just scientific research that is not involved with, you know, with virology or just COVID-19 in general, did take a hit.
Mhm.
So for the, during the first lockdown, let's say, yeah, we were all of us were at home and trying to work from home. But obviously with scientific research, it's not the same thing. Of course, you still want to work with animals. You still need your specialist equipment, of course, to do different things. So yeah, a lot of our work took a hit, I would say, but mine didn't, because I was still mostly computational, as I said. I kind of, it was essentially, I was doing the same thing but from home, and of course without the human interaction that I've loved so much in
Yeah.
the lab. So we've been, I mean, touch wood, things are getting better, and I have been going back to work, you know, most days, you know, for most days of the week, since beginning of March. So things are kind of looking like they're back to normal,
Okay,
but
yeah.
yeah, I'm not gonna say that. I'm not gonna jinx it. So that's typical day, let's say.
So you said you do computational work. What specifically are you working on at the moment?
Okay, I have two main projects, and I can try to briefly describe them. I am involved in, first of all, okay, I'm going to call it the clustering project, and you will understand why later. And I'm working on this project with a postdoc in the lab. And essentially, we know from previous research that there are specific neurons in the hypothalamus of the mouse brain. So the hypothalamus is kind of the control center for, let's say, innate behaviors. So behaviors that you don't really think about, because a lot of hormones are actually taken in the hypothalamus, and the things such as, like, you know, digestion, feeding, aggression, stuff like that, in animals at least, they don't think about, they just know to do them. And we're really interested in kind of breaking down the circuits underlying these behaviors. So back to the hypothalamus. So we are one of the behaviors we're focusing on is parental behavior. So, you know, a mouse gives birth, has some pups. She knows how to take care of them straight away, you know. And we're trying to, you know, break it down and figure out, you know, how does this happen? How does, you know, how does that behavior come about? And from previous study, we know that there are specific neurons in the hypothalamus that have this protein called galanin. And they have been shown to be the main population of neurons that are actually activated when these parental behaviors are done by a mouse.
Wait,
So
so sorry, how does the
yeah,
protein relate to the neuron?
so essentially there are I don't know how many millions or even billions of neurons in the brain. They all have different characteristics, and one of the main ways that we can kind of differentiate them is through the proteins they have, like on their cell membranes or inside their cell body. They all have differences in, let's say, their DNA that does end up in them producing different proteins, right? Because DNA gets converted to RNA, then gets converted to protein. So that's how they outwardly show that they're different from one another, right? So how we detected that, how we know that these neurons have galanin, that's a story for another day. But we just know that these neurons have been activated by parental behavior, and we then found out, oh, these neurons mostly have galanin. They mostly produce galanin. So we're like, okay, interesting. And so from there, we decided to essentially delve a bit deeper into this population of galanin neurons in the hypothalamus. And we're trying to find out if they can be further characterized by, like they can be further split into subgroups that maybe are defined by the way they fire, because neurons are they have electrical properties, right? So maybe there are different subgroups in that galanin population that are different in that way, or maybe they are different in their morphology. So we kind of delve deeper.
So by morphology, what does
Yeah,
that mean?
let's say neurons can have many different shapes. They can have like fat cell bodies, small cell bodies. They can have very sort of dendrites that are very long and very far reaching, and they can have just like their dendrites, for example, can be thicker or thinner, or they can have some things we call spines sticking out of them that essentially are where the other neurons can connect to it. So they have these different properties that we can characterize by modeling them. And in terms of the electrophysical properties, I can give you a few examples. So we can measure firing frequency, we can measure action potential amplitude, right? Because they have action potentials. Depolarized the neuron gets also depends on the neuron and its properties. We can measure membrane potential, membrane capacitance, all of these sort of electrical properties of these neurons, and we can track these properties as well as the morphological properties. And we're with these two different sets of parameters from each from these neurons, we can then perform this is where I come in. I then perform unsupervised clustering on this set of data. And this unsupervised clustering, what it does essentially is, unsupervised means that there is no input from me, there's no input from anyone in the algorithm, but the algorithm essentially tries to find trends in this data. So this data being electrophysical sort of parameters and the morphological parameters. So if we input them into a clustering algorithm, it's gonna essentially try to guess at if there are any similarities between these different neurons just through these parameters. So if a lot of neurons have a low firing frequency with like a small action potential amplitude, then okay, they are very similar. They kind of the algorithm would kind of bunch them together. And from that, we can then, of course, we can't rely solely on the algorithm, and we can then, it helps us out, and it kind of would point us in the right direction in trying to find if these neurons are indeed part of a subgroup or not, or it could just be sort of, they're just all different. But we're trying to see if we can find different populations within these galanin neurons.
So different like brain functions that like we do or we have, they are done by different like types of neurons.
Yes, yeah.
Okay.
Essentially, they are done by different types of neurons being connected in different ways.
Okay.
So there's also that connection as well between these populations that does kind of end up forming a behavior, an external behavior or like a thought. So it's essentially, the field of neuroscience, well, more specifically, the field of, I guess, connectomics does look at, let's say, systems neuroscience looks at these different populations and how they're connected to each other and how that leads to something, how
Okay.
that leads to, a thought or a behavior or sort of anything that's outwardly expressed. So yeah,
Okay.
it's quite complicated in there, and we're trying to break it down.
So your algorithm, what it does is, so are you taking data from what, like these signals that you've gathered,
Measure,
and
yeah.
then the algorithm separates these into different groups by just
Yeah, just by looking at the data, because let's say if there's only two parameters that you're measuring, for example, you can kind of plot them in a 2D graph, right? So it's
Mhm,
like, oh, you have, like, let's say, action potential amplitude on the x-axis, and let's take a morphological parameter, let's say cell soma volume on the y-axis, right?
mhm,
So if you have, like, well, then you'll have a correlation, right? You can have maybe a lot of cells that have a big cell body, and whose action potential amplitude is a bit lower, and then
mhm,
maybe there's like another group that would have like a smaller cell body but a higher action potential amplitude. So you could actually see this. You can actually maybe see this trend on a 2D graph, right? If you do look at two different parameters only. And the thing is, yeah, so that can maybe be done by a human. You can be like, oh, hold on, there's like, seems like there's a trend here. There's two different groups that kind of have similar properties here. But the more you add parameters, because we have a lot of, I don't know how many we have, but we have a lot of different parameters that we look at. So that's just going to be if you think about it, you can kind of plot the data in like XD, in like this XD dimension, that then you would see these different clusters if they exist.
mhm.
But since we can't really do that, I mean, you can't really visualize that
Yeah,
as a
over
human, but like
over
the
3D
algorithm
dimensions. Yeah.
can kind of like somehow kind of sort it out for you, essentially, and tell you if there's something there. The more dimensions your data has, the harder it gets for someone to kind of interpret it. You would then go on to using an algorithm to kind of do that for you, because coding is amazing and helps you out in essentially interpreting your data.
Did you come across any problems with this algorithm, or like how do you know that your algorithm is right
Is
for what you're
working?
trying to do? Yeah.
Well, it's actually, so I'm using Python as a programming language, and there's actually quite a lot of different libraries that you can use, and these libraries have different functions that do different things. And Python is quite, I don't know if it's the most popular programming language, but it does have a lot of machine learning and sort of data analysis libraries that are well documented, and you do and they have these functions that kind of already coded out for you. And the main thing you have to do is understand how these functions work, what to input in that function, what it outputs. And a lot of that is actually explained in the library's documentation, which is I guess written by these experts that coded the function in the first place. So it's not too hard to figure out what to do. And essentially, if you use the function correctly, then you'll know, okay, yeah, this I'm doing things correctly, this makes sense.
But how do you know it's correct?
So I mean, if the function isn't, well, you essentially just trust the function to do it for you. And if the output doesn't make sense, then it could just be because there's no trend there, or maybe you should do a bit more pre-processing on the data, and let's say cut out some noise, because maybe a lot of these parameters are similar to each other. Some of them are similar to each other. So adding similar parameters to the algorithm, let's say, would be like adding some unnecessary data. So you could do some pre-processing there and kind of narrow down the parameters into like the essential ones that actually show some variance between each other, right? And then again, the output would be what you get essentially. And you can essentially measure if the clusters you get, right, because you essentially, if there is a cluster, then they will tell you, oh, okay, there's, let's say, 4 different clusters of neurons from these parameters. Then you can also measure parameters from these clusters that essentially tell you how bunched up these clusters are, or if they're quite spread out, then that's not really a cluster. So you can do some, I guess, analysis after the clustering is done to essentially tell you if this cluster is a good one or not. And then in the end, it all comes down to you deciding whether or not this cluster is actually like a real cluster or not, just from I guess analyzing the data yourself, I get after the clustering is done, or just like I think it's the clustering is there as a guide, but then you definitely at least in the scientific field, you definitely have to kind of look back at, let's say, these clusters and the parameters from these neurons and kind of like investigate yourself, and then you're like, actually, yeah, this makes sense. So then you'll be like, okay, yeah, there is a trend there. This could be a new subpopulation of neuron that we can investigate further. You never know whether it's like it's never going to be completely 100% yes, this is the right answer. But this mainly guides us towards further exploration. So you cannot, I guess, you cannot take the output as final answer. This is it. That's at least for machine learning stuff in science, in the scientific field. But then more concrete things that you can like just rely on the algorithm by itself. But for scientific research, it's just they're like, yeah, they're just a guide, let's say. So that's that project. But I do have another one that I don't know if you want me to talk about.
Yeah, no, please go ahead.
Yeah. Okay. So, so there's the clustering project. I call it the clustering project because, as you guys know, essentially I use an algorithm to try to cluster this data, right? And the second project essentially involves using MRI essentially as modality to investigate whether we can find any structural changes in the brain during pregnancy or during/after pregnancy. So again, this is another project that's focusing on parental behavior, as the one before, but we're looking at the entire brain instead of looking at just this one population of gallina neurons and try to kind of look more into that. We're actually just looking at the whole brain through MRI scans, and essentially, they're ex vivo MRI scans. So essentially, mice are anesthetized, called. We do ex vivo brain scans of some virgin mice and brain scans of mice during pregnancy. And we have two different time points for that. We have, let's say, midway through a mouse's pregnancy. A mouse's pregnancy is around 21 days. And
very short.
so, yeah, I know it's quite different from humans. You cannot compare. So let's say we extract some brains and we scan them at day 10 of pregnancy, and then further ahead we scan them at day 18 of pregnancy. So let's say day 10 is like a midway point of pregnancy, and day 18 is like late pregnancy. It can be considered late pregnancy. So essentially we have, let's say, this, and then we do have some postpartum brains. So some brains after the mouse gave birth. So we have these different groups of brains that we've scanned throughout the year, I guess, throughout this past year. And where I come in is I'm trying to use some image analysis sort of methods to figure out whether we can find some sort of changes in morphology in these brains by comparing these different groups to one another. So the method I use is called deformation-based morphometry. So we'll break it down. Morphometry just means morphometrics. I don't know how to say it properly, but it's just essentially the measurements of morphology. So morphology being, the shape, size, structure of something. And deformation based, I will come to deformation and why that's important in our pipeline. So what we first have to do with these groups of brains is essentially we have to kind of normalize them, in that we have to, they're all kind of slightly different, right? They are going to have slightly different changes in their, like they're going to have different structural sort of, they're going to have different structures and different sizes and stuff like that. So we're trying to, we want to look at these differences, but how do we do that? How do we extract these differences? So the key here is image registration. So let's say, okay, registering an image. I'm trying to see if I can find like some sort of example. Okay, so I have two hair bands here, right? So let's say this is one image and this is the other, right? It's a bit like wonky. And essentially, imagine these are like images of circles. So a registration would essentially, if you want to register this circle onto this smaller one, what you would have to do is, registering essentially means you want to overlay them perfectly onto each other. So as you can imagine, if this circle will have to shrink quite a bit to then, I guess, be registered to the the first circle, right?
Mm-hmm.
So you can see, and I guess this is the same for brains, except that it's a bit more complicated. Brains have a bit more detail than just being a circle. They have, if you look at an MRI scan, they have kind of sort of different intensities in different areas of the brain. You know, some white matter, gray matter, and they have like outlines of different areas. So essentially, we're trying to match our brains onto, this is going to be this average brain, this reference brain that can be created by us, or it can be created by any sort of other institutes. And essentially it's like an average of many different mouse brains that we can kind of take as a population average, right?
Mm-hmm.
And so registering our brains onto this template slash reference brain, we're actually essentially, so our brains are going to be a bit different, right, than this average. So by registering one of our brains onto the average, we essentially have to deform the brain so that it matches perfectly onto our reference brain. And that's where I guess deformation based comes in. These deformations needed in, let's say, our image registration is what we then look at, because that is kind of where the differences between these two brains are, I guess, found. As like that's the output of a registration. You'll get these deformation matrices.
Sorry, so in terms of your project, because you're working with mice, what is the reference image? Where are you getting that from? And what are you using as reference landmarks or points to match them onto, like match images to each other?
Yeah, it's true that you do have to look at reference, landmarks in these brains to kind of match them perfectly onto each other. So to answer that question, I don't exactly know how this matching happens, because it's not done by me. It's done by, I use this library that is coded by experts in the field, and they actually coded this function that maybe looks at, let's say, pixel intensities and tries to match them in that way, or maybe looks at peaks and trough in the image. And I don't know exactly what exactly the registration looks at in terms of landmarks, but it's mainly going to be either landmark based, so maybe looking at these specific areas in the brain and matching them there, or it can be just looking at the intensity of a pixel and trying to match, I guess, the intensity pixel by pixel. Your second question being the templates, the reference brain, where do we get it from? Or so we've been playing around with it actually recently. We've tried creating it ourselves by averaging a bunch of our own brains together, mostly I guess virgin, right, for it to be the average population brain of just like normal mice. But the problem with that is that essentially it is better statistically to exclude, essentially, if you create this template with these brains, you then have to actually discard them. You cannot use them later on in the pipeline to then compare to let's say pregnant brains, because then they can be there are going to be more similarities between the templates and these brains than any other brain that's, I guess, extracted from any other mouse. So then are currently actually looking at using a template brain, a reference brain created by another institute, and it's quite a big institute in terms of neuroscience, and it's called the Allen Institute I think they're based in Seattle, Washington, but I'm not too sure. So they essentially created this average brain from the same strain of mice that we're using, and they've used a lot of mouse brains, so it kind of saves us from doing it ourselves. So
wait, so sorry, I didn't
we
understand
have been using both the same.
why the ones that the reference brain that your like lab had created was not sufficient. So
it's essentially, no, I think it'll be fine, but we essentially have to, because if you do, let's say we have as I said, we have different groups of scanned brains. We have like some virgin ones, some day 10 pregnancy ones, some day 18 pregnancy ones, right? And if we use all of our virgin brains, let's say, to create this template, because essentially this template should have at least, like, the literature kind of standard is around like 20 brains per template, then we would just use all of our virgin brains to create it. And let's say, if you think about it, if you have an average of these 20 virgin brains and then you register, because you still have to register, we still want to register, all of our brains to the templates. But the problem is, if you register our virgin brains onto our virgin templates, you'll have a lot more similarity
Okay, I see, I see.
between the virgin brains and the virgin template than anything else. So it skews the data. It skews the
Okay,
data
yeah,
towards favoring the virgin. Essentially, it'll be like, okay, right, there are essentially no differences between these virgin brains and the virgin template.
yeah, okay.
Fine,
I
but
see.
it does skew the data a bit towards,
There being no
preferring virgin
differences,
brain.
yeah.
And I mean, the way to fix that is essentially to scan more brains and just use them to create a template and not and exclude them from the analysis later. So we can do that. We're essentially planning on scanning more brains, but for
Hmm.
now, that's the issue we have.
But
Yeah.
could you still not use the one that you created with the virgin brains when you're comparing the like 18-day one post pregnancy one?
That's true. You can. Yeah, and that's something that we will do in the future for sure. But what we've been focusing on so far, I guess, is because there's a lot of comparisons you can do, right? You can compare
Hmm.
day 10 to day 18 to postpartum, day 10 to postpartum. But I've mainly been focusing on comparing virgins to late pregnancy, because late pregnancy is essentially the time point at which we see the most changes in behavior. So the mouse becomes a lot more maternal, and it's nest building, because essentially they build kind of this nest from some hay, not hay, but similar
Hmm.
kind of material. So they build these nests for their pups, and like the nests are like so much nicer, and yeah, you do see the most changes in behavior around that time point. So that's kind of why we want to focus on that one, and why we think there might be the most changes in the brain structure at that time point. So and comparing that to virgin would kind of show the most contrast. So that's kind of the main comparison that we're looking at right now. But you're right in saying that we can use our virgin brains that we currently have to compare any other groups that doesn't involve virgins, I guess. Yes. So anyway, so these registrations to the template, right? As you can kind of tell from my terrible demonstration,
No,
there
it's
is
pretty
going to be
good.
some deformation, right? That is needed to kind of match them perfectly onto our templates. And yeah, we get essentially these differences from these deformation matrices. And these deformation matrices is essentially what we get them for each brain. So you'll get a bunch of them in different groups, essentially. And we then we essentially compare these different deformation matrices to each other. And to do that, we have to model the data, because how do you compare a bunch of deformation matrices to each other? Essentially, you model one group for it to kind of for it to be like, oh, that's an average deformation matrix, and then for virgin, and then that's an average deformation matrix for late pregnancy, and then you can then compare them using statistical analysis, such as like t-test or like ANOVA. And like these are kind of statistical tests that like are very common in the scientific world that I kind of don't really want to get into, but like so t-test is actually one of the main ones, where you just compare. A t-test is essentially comparing the mean. Let's say there is going to be a mean deformation matrix for virgin and a mean deformation matrix for the 18, right? And you compare, essentially you do one mean minus the other, and then you standardize them by dividing by standard error or something like that. And then from that, and that t-value is going to show you kind of the differences between these two groups. And then you can I guess, do statistical inference, which essentially involves looking at where exactly these t-values are actually significant. So I don't know if you guys remember anything from sort of high school. You have like, if a t-distribution is usually kind of bell-shaped curve, like has kind of this bell shape. And when you do want to look at significance, you actually look at the t-values that are outside the norm being where most of the t-values are, right, at this bell shape. So if you want to perform statistical inference, you actually look at the ends of the distribution, and then you're like, essentially, there's like the 95th percent, or the 95th percentile is where there's only 5% of t-values that have this value, which is I guess quite different from the norm. So that's kind of what you would look at to say, oh, right, these t-values are quite significant. They're quite different from I guess like our mean of 0, let's say.
So are you
Yeah,
looking at how many t-values are away from this mean? Or
essentially, and essentially, our mean is going to be at 0, actually, because our deformation matrix. Essentially, we log our deformation matrix so that any value above 0 is going to be okay. I didn't explain that. I didn't explain the deformation matrix step too well, but essentially these deformation matrices, you can kind of think of them as vectors, right? So let's say, you know, the bigger circle is going to shrink, but it's going to shrink like, I guess if you think of the circle, like the arrows are going to go inwards, right? To shrink it if you think of it as a vector. So you essentially, this deformation field is actually then refined in that we just look at, since we are looking at images, these images are going to have pixels and 3D images are going to have voxels. And we essentially refine this deformation matrix to tell us this voxel was shrunk during this registration. This voxel was expanded during this registration, because again, they try to match the brain's voxel by voxel. And that's kind of what we want to look at, the shrinkage or the expansion of voxels that happen during a registration to the template.
Sorry, what do you mean by voxel?
Voxel is like a 3D pixel. So if you think of a pixel is like, right, a square in 2D, right? Like, you know, every image is going to have thousands of pixels, but a voxel actually has depth to it because it's like a 3D image, right? So you can think of a 3D image as a bunch of cubes, and each of these cubes is going to have some sort of color, right? So a lot of voxels is going to give you a 3D image essentially that you can look through. It's essentially a pixel with volume. And yeah, essentially these. So we then kind of extract the information that tells us, is this voxel shrunk or is this voxel expanded? And we then essentially log these scalar values essentially to then show us if a voxel has expanded, then the value for it is going to be bigger than 0, and if the voxel has shrunk, then the value associated to it is going to be negative, so it's, you know, lower than 0. So you can think of, if you know that these scalar values, let's call these scalar values, they're technically determinants of the Jacobian matrix, of the Jacobian of the determinant matrix. But yeah, that's kind of some maths. If you look at, again, back to our virgin versus the 18 of pregnancy example, if the t-value, which is the difference of the means, is going to be around 0, then that means that there has been no expansion and no shrinkage at that voxel, right? Because the difference there is no difference between them, no difference, right?
Yeah.
So that's kind of like, that's going to be so a lot of, I mean, you're going to have a bell-shaped t distribution that is going to be centered around 0, is what I'm trying to say.
Mm-hmm.
And yeah, any of these extreme values are, which we will then, I guess, detect by statistical inference and p-values and stuff like that, p-value of 0.05. These are the ones that are like really interesting because they differ from that's where there's either a lot of expansion or a lot of shrinkage in the brain. Yeah. When you compare these different groups, you can compare. Yeah, you can compare, again, more than virgin versus day 18, but that's kind of what we're focusing on. And yeah, and these I guess these expansions or these shrinkages will then tell us, oh yeah, there is a kind of a significant change in morphology in the brain at that area, because again, we have a 3D map of these t-values. So from there, the goal is to then use this information, use this, I guess, statistical analysis to then kind of explore further into, let's say, that area of the brain that has been expanding. And we then have to figure out why is it expanding? Is this something we can see with looking at the brain in different ways rather than just MRI, etc., etc. So yeah, it's a good, I guess, essentially this analysis will then kind of point us to different directions and different projects and different I guess questions as well. I feel like I talked like for ages, but that's like project number 2 is this MRI project. And yeah, I hope I made it clear. But,
so where or how are you obtaining these 3D images of neurons?
For
like
neurons, as in for the clustering project, no?
For the second one, for the, I think.
So the brains, you mean, right?
Yeah. So the pictures of brains, like how are you?
Yeah. Oh, it's just essentially an MRI machine it's really cool. It's just an MRI machine, but like it's smaller. So you essentially fit you fit the brains into this cylinder, and then you just stick it into the MRI machine, and then it just images it as an MRI machine does. And then you essentially get the scans. Yeah, you kind of get the scans in the end, and then that's where I come in and I kind of look at them and stuff. Yeah, it's simple MRI machine, but tailored to smaller samples. So obviously, like a human MRI machine is going to
MRI
be a
is
lot.
huge, yeah.
Again, it's like technically, yeah, this scientific equipment is just very technical, and we kind of rely on it a lot. And yeah, that's why things like lockdown kind of slowed us down a lot, because we just don't have access to that through home. So yeah.
And for the clustering project, how
Yeah.
do you obtain images?
So for the MRI, for the clustering project, again, we're looking at things ex vivo. So we obtain the brains and we actually slice them coronally to then actually, so as I said, a postdoc in our lab looks at the biophysical, the the electrical properties of the neurons. And how she does that is essentially it's this process called patch clamping. So patch clamping a neuron, essentially you kind of you stick let's say a needle into the neuron, and you excite it with some current, and then you actually also measure its response with, like, let's say with some sort of electrode, right? That also is in contact with the neuron. So you first get slices of the brain, and these slices are of course gonna have the hypothalamus in it. So the hypothalamus is only a certain region of the brain. So we slice the brain until we get to that region, and that's the slice we look at. So once our postdoc kind of finishes with her electrophysical analysis, why do I keep saying that? With her, like, let's say biophysical analyses of sort of measuring the electrical properties of the neuron, she then images these neurons using this a confocal microscope. And it's quite a complicated microscope, but essentially what it allows you to do is, it's a very thin slice of tissue, but it actually allows you to image different, let's say if this is a slice. I mean, this is going to be the brain, but then if you put it on the slide that you're going to image, then you're going to have, since it does have a thickness, you're going to have different layers. You can actually image different layers of the slice by focusing in on different layers, essentially. So you can first image the very top of the slice, and you image like just a bit below, and then just a bit below, and just a bit below. You can essentially end up with a stack of images.
Okay.
So that allows you to kind of have, obviously, the X and the 2D image, but have also a Z component to it. So like a depth component to it. So you end up actually seeing this neuron, because of course, like, that's the whole slice, but she actually focuses the microscope on this one neuron that she recorded from, and you can actually see the its dendrites and its axon, and just through imaging it confocally. So in with these different slices, so you end up kind of looking at a 3D image of the neuron, and it's really cool because you can just see it like branching out and stuff. It's really cool. So that's kind of how you image the neurons, to then later on look at them and then get the morphological parameters that I mentioned before, such as cell soma, and like how many dendrites and how much the dendrites branch out and stuff like that. I try to make it clear, but again, confocal microscopes are quite complicated, but you can have a look at, I guess, online and stuff, but in terms of exactly how they work. But essentially they allow you to kind of measure in the depth of a tissue, as well as obviously just looking at it in 2D. So yeah.
Would you say you work more in like experimental biology or computational biology?
So I'm mostly computational, but of course I've just by being in the lab, I've learned a lot of experimental techniques that most of, I guess, the neuroscience field uses. Or just a lot of it is going to be done by many different labs in the same way. There are different techniques. So for example, patch stamping of neuron is quite a common thing to do. Using a confocal microscope is quite a common thing to do. Using MRI machines, a lot of different labs use them. So I end up learning a lot from just people doing these things in the lab, and then you're like, oh, okay, so then you understand what everyone else is doing at the same time, because you're like, oh, so they use a method that our postdoc is actually using. And so when you do read more about different research in different labs, you're like, I get this now. It's
Mhm.
cool, because you end up learning from your own lab, and and the experimental techniques used in your lab. So I'm computational, but I learn a lot of experimental stuff, and I'm interested in it as well. So yeah.
So since you did engineering first, and then you went into kind of neuroscience, what
Yeah.
was what challenges did you have moving like to a different
Moving
field
into it.
essentially?
Okay. So first, for sure, just like learning these, as you just mentioned, like these experimental methods, there's quite a lot of different things you can do with, like, with tissue, and how to image it, and how to measure from a neuron, and also, of course, there's like, you can activate a neuron with using like lights. That's like the whole field of optogenetics. And
Mhm.
there's quite a lot of different methods used in science, and of course there's like genetic tools that allow us to target these neurons with different proteins, let's say. So it was a lot to learn. So I think my main challenge is essentially just figuring out what was going on, how the heck they measure that, the different techniques used is essentially like the main thing that I was struggling with. Like, okay, I'm like, okay, so they use this genetically modified mouse, and then they targeted these neurons using they essentially inject viruses inside the brain of a mouse to then target these specific neurons. And the virus knows how to target these neurons because the mouse has been genetically modified to then, let's say, express some sort of protein in these neurons only. And it's like, okay, yeah, it was quite a lot to understand. And the main challenge was, yeah, reading more about these methods and understanding them, so that I could then like keep up with what was going on in the lab, I would say.
So what maybe tips or advice would you have for someone who had the same kind of idea, like to move into this kind of
Yeah.
field?
So essentially, something I didn't do before too much, just as in an engineering degree, you don't think about it that much, but in science, it's very important. It's like just reading papers, reading scientific papers, and especially like reading, like, how a paper, an article, however you want to call it, has like, intro, results, methods, results, discussion, conclusion, right? Yeah, the interesting part isn't the results, but if you do want to go into a field, so read a lot, and into a specific lab, read a lot of their papers, and what exactly they're doing, and just also focus a lot on the methods section, which is the section where they're going to explain how they do things.
Mhm.
That's the main thing that you should kind of read a lot. Yeah, you should read a lot of papers, and you should also understand how exactly they do things. So yeah, a lot of reading essentially is key for you to kind of keep up with the scientific community if you do come from engineering, that's for sure. Yeah, that's the main thing I would say to do. Read papers.
Because you did engineering, what unique insights did you bring in like to your lab, like your current lab with your engineering experience?
So I would say, I mean, I recently graduated. I don't think I had that much experience with like things, but I think the main thing I do add to the lab is obviously my coding ability. Let's say I do know a lot just from, I guess, my degree. I do know a lot about just coding in general, and I think that's the main thing I bring to the lab, just my knowledge of coding and algorithms and stuff like that I then apply to these data analysis pipelines, as I've just tried to describe, with my two projects that I'm involved in. So yeah, mainly definitely software. I guess my software knowledge is kind of what I bring to the table in the lab.
Mhm. And was most of this like software knowledge stuff you gained from stuff you had to do in your degree, or did you ever do like extra projects or extra work that contributed to like the knowledge you have now, essentially?
So it was mainly, I would say, from my degree. So and of course, with an engineering degree, I did have quite a bit of practical work. So as you, well, maybe not during COVID, but you should be having sort of group projects, or even
Mhm.
like, yeah, group projects in engineering, like every year, or
Yeah.
in your last year it might just be individual, where you just literally are like yeah, and you try to make something from scratch, or you try to develop this new product as an engineer, and that's kind of that's where you learn the most. So it's not going to be in the lectures, not so much. I mean, you do have programming, let's say, modules that you do have kind of like practical aspect of it, where you go to a computer room and you just code different things and you have assignments. But it's mainly going to be in projects that you learn a lot more about coding than anywhere else, really. I mostly learned from my projects at uni, and I also Python was a thing I learned myself at the beginning, and then I used it as a language for my final project at uni. So I definitely did have to learn by myself. I did self-teach myself, I guess, Python, but that was actually a lot easier. If you do know different languages, it's actually a lot easier to learn a new one.
Mm-hmm.
And I knew MATLAB from uni mainly. We mainly focus on MATLAB at uni, and we did a bit of C, C++. So I think it's uni obviously helped the most, but I also had to I had to self-teach myself Python, at least. That is now the main language I use. So yeah, it's mainly uni, but I also did things on the side to help out for sure.
So what kind of things were you doing on the side? Was it kind of there's like website like learning
Yeah.
from websites or?
Yeah, essentially, you have a lot of different resources. The website I used was, I mean, I used a bit of, I did, yeah, there were two websites that I kind of focused on. One of them was like Data Camp, and the other one was Code Academy, I believe. They do have like free sort of intro to Python, or some sort of course
Yeah.
that had that name. And I kind of used those, and then and then the Python kind of got better with my projects at Imperial. So yeah, I didn't do too much. Let's say I didn't delve too much into courses online, because I was like, I mean, I was still, at uni, and I was doing different things, but these intro to courses are actually quite useful for if you want to start up, if you want to learn a new language. So I recommend kind of looking at these websites like Code Academy, etc. They do have quite decent free courses. So yeah.
So what would you say has been the coolest project you've worked on, coding or otherwise?
I mean, it has to be what I'm doing now.
Mm-hmm.
I would say so. Yeah, because I think I would say, I mean, both. I don't think I can choose between the two projects, to be honest, that I'm currently working on. So either MRI or clustering. They're both really cool. But I would say, I would go for the MRI, because it's just because of the things I've learned from it. All of these different image processing methods. So like image registration, like deformation based morphometry, all of that. It was really challenging to learn that from scratch, essentially, because there was no one else in the lab that knew about image processing or MRI or anything like that. So I'd say that project has been the most rewarding so far, because of the amount of effort I've put in it, and also obviously the output is going to be I guess the result of that project is actually going to be really cool, and I'm going to have hopefully a paper published with that data in it. So I'd say that the MRI project is my most rewarding so far.
So with those projects you're currently working on, what do you think are the wider implications of like the results you think you might get?
So I mean, I can't really tell you right now what we're gonna get from it, because again, we don't really know the results yet. But we're essentially the aim is us trying to understand. In my projects, we're trying to understand pregnancy better, and how that affects the brain, and maternal behavior in general. So I guess just in the long-term implications of these projects is that we will understand exactly what goes on when you become pregnant, and if there are like changes in the brain circuits. Are these like permanent? What, let's say we can then let's say try to translate our kind of knowledge of that's a mouse brain, because that's still what we're looking at. We can then kind of be like, oh, okay, this could be similar in humans, because they're both mammals, right? Mouse and humans are mammals, so it's not that much different. Like our genetic makeup is not that much different, so we could maybe get some insights into the human brain and pregnancy, and maybe what's the long-term implications of pregnancy are. And yeah, I think it's, I mean, if you're not that interested in maternal behavior, then that it doesn't seem that cool. But is it in neuroscience, it's all about understanding the brain better. So if we have that insight there, then that's already an achievement, I would say. But yeah, our work is not as translational as what you can find in other labs.
Mhm.
But
Okay.
it's mainly going to be, the goal is mainly going to be, pushing our so furthering our understanding of the brain and of these different regions of the brain in different circumstances. So like, let's say in maternal behavior, pregnancy, etc., different behaviors. I would say it's not going to be like, it's not going to turn into a product or anything, but it's mainly just going to be the scientific community is just going to learn more from the work we do now, essentially. Yeah.
A little bit of a different topic now. Have you faced any kind of challenges or adversity because of who you are or your background, or like how you might identify yourself in the field, like being a minority in the
Yeah.
field, do you think?
I mean, I think I'm lucky enough not to have faced any adversity in terms of, so, I mean, what I'm thinking of right now is just applying for, I mean, this is my first job, post uni. So in terms of applying for this job, I didn't actually face any adversity. And even I'm thinking of uni and I'm like, yeah, no, I don't think I was negatively affected by any by, let's say, being a woman, or, being a woman of color or anything like that. Well, but I say that you actually, like, you never actually know, because I
Mm.
mean, but then again, I've only applied for one job, so I can't tell you. Like,
Yeah.
I've only, had one job so far. So whenever I do apply for different jobs and I get like rejected because of some unknown reason, then I'll be like, okay, interesting. But for now, no, I think it's been, I've been, let's say, treated as equally as I should be, let's say. Yeah.
That's good
Was
to
that your
hear.
question? I'm not even
That
sure.
that was my question. I was
Okay,
just interested
cool.
if you had
Yeah,
experienced
yeah, yeah.
anything like
For
that.
now, no.
That's
So
good.
far.
I'm
But
glad to hear it.
and that's, I'm still young, so
Yeah.
we'll see. Maybe,
Mm.
hopefully not.
And for people who don't know, I have the privilege of playing, or I have played in the same
Yeah.
football team as Estelle. So, playing football, well, for me personally, I've faced a lot of, I wouldn't say
Mhm.
adversity, but kind of like an expectation because I am a girl. And then when
Oh
it
my
comes to
God.
football, which
Yeah.
is a male-dominated sport, so
Yeah. Yeah.
what impact have you had any experiences, and what kind of impact has football made on your life?
In terms of football, like, there's always, even any, to be honest, most sports, it's so annoying, because whenever someone sees, let's say, a woman play football, especially in the UK, it's like the biggest sport, right?
Hmm.
They're like, they either, underestimate you, or
Hmm.
they, you do get like some of these like unnecessary, they just come to you and they're like, oh, cool, you play football and stuff, and they just assume things about you, that, like, just from you being a girl playing football, and it's just these prejudices, I guess. And yeah, I've faced that a lot in just playing football in London, throughout, I guess, the past years. How has that affected me? I would say I always like to prove them wrong. So always, like, what that has done is essentially just push me to kind of become better, and kind of not silence the haters, but essentially, prove to them that a woman playing football can be just as good as a man playing football, if not, I mean, of course, there are going to be some differences, but like, you always want to, you kind of push yourself to be like, you can't
Mhm.
tell me nothing,
Yes.
it applies to any sport, to be honest. Like, I don't know why women playing sports it's become, obviously, like, more women play sports now, but maybe back in the day, in our parents' day, maybe it wasn't that much of a thing. And I think it's getting better. The perception of women in sports is just getting better. But yeah, it's pushed me to kind of become better at sports, and anything I do, actually, because
Hmm.
you will also get these prejudices at work as well. And
Yeah.
it's always, you always essentially push yourself further, and you always want to be kind of on top of things. And also, like, let's say, if you want to explain something, or let's say, if I want to give a presentation of my work, today, none including, but like, I always make sure that, I know everything. I know, like, let's say, I know what I'm doing, and I know everything I want to say, and I can answer any question, etc., etc. So you always, like, you're always hyper prepared, I guess, as a woman, just in general, like just so men don't tear you down, let's say. Yeah.
Having to do this extra preparation puts a pressure on you. So how do
Yeah.
you deal with that pressure, and or anything you do to
Kind of.
not get stressed about it? If that makes
Yeah, of course, man. Any type of work is going to be stressful, right? I mean, it helps a lot to do sports, I would say. Like, just if you have a busy day at work, if you have a stressful day at work, my main relief is sports, actually. Yeah. So football. I also like do cycling. Yeah, it's a good way to mentally kind of stay sane. Let's say if you do have
Yeah.
an extremely stressful week at work, and you still have kind of the sports to look forward to, right? So essentially I cannot live without sports, and I recommend anyone to find, well, it doesn't have to be sport, but find anything that kind of takes your mind off of work for a while, that's really important. And that's essentially what helps me in coping with stress, and yeah, all of that extra stress that I would have as well, which doesn't, let's say, it's mainly for presentations, and also stress in general from anything. You can definitely get a relief from that from with sports or with any kind of hobby that you love. So I recommend definitely doing something else other than work, finding a hobby, finding that hobby or sport or anything. But it's really important, I think.
On presentations, or what kind of tips would you have for someone who might struggle with presenting, or especially in an academic setting and in a science kind of setting?
Yeah, I still don't think I'm great at presentations. I kind of get the stage fright. It's
Mhm.
weird. Even in Zoom, even like,
Yeah, no,
'cause things
it's
have
me
gone to
too.
Zoom, and I'm still stressed. Advice. Okay, so if you want to present, I do have like my PI that I can kind of go to ask for help, ask for. Okay, so first of all, plan the structure of your presentation. That's for sure. And then maybe, yeah, go ask your boss or anyone who kind of could help you. Go ask them sort of their opinion about, like, okay, the structure of your presentation, or do they think it looks all right. And then you can kind of, if the structure is there, that's the main thing. If the structure is there, so let's say you want to focus on this part of, let's say I want to focus on this part of my MRI pipeline for this much time, and then I move on to this, and I move on to that. If you know exactly how the presentation is going to go, then that helps a lot. And also, don't be afraid, because honestly, I've done it before, but don't be afraid to kind of write down, like, not exactly what you want to say, but just write down kind of bullet points of exactly what you kind of, the points you want to make for each slide of a presentation, let's say. And another thing is just practice, keep practicing, I guess. Practice in front of someone, even like
Yeah.
like a flatmate or anyone you can find that's an actual person, because it helps. So I would say, get the general structure of your presentation kind of right, and don't be afraid of writing down bullet points in practice, really. And also, as a procrastinator, I would say don't leave it too late as well. That's a thing that I used to do at uni, at least, but now I'm like a lot better in terms of leaving myself enough time to kind of like fine tweaking the presentation, like making the figures nice, and the spacing nice and all of that. But yeah, that's I think that it might just be me, but I'm just like super I'm such a perfectionist about these things. I don't know.
Yeah, no, I definitely agree with practice. I mean,
For sure.
just
Just like
even the most
say it out
random
loud,
like presentations.
essentially.
Yeah.
Yeah.
Kind of putting yourself up to do it when there's one person, like just offer yourself to practice presentation essentially.
Yes. 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 Yes.
Who is your role model, as in maybe when you were growing up, and has that changed since?
I mean, I do admire quite a few people, but I wouldn't call them my role models. I mean, it's a bit cheesy, but the main one I would say is like, oh, my mom, like,
Yeah.
yay. Yeah, she's been the one to like push me. Like, she's kind of successful in her own way, and she's pushed me to really go for, I guess, to pursue this career in STEM. Actually, she's like, yeah, why don't you? Because I didn't even think of engineering until my last year of high school, even, or like, whenever I was starting to apply, and I was like, let me do science. But actually, like, when we went to these open days, and my mom was with me, and she's like, why don't you do engineering? Doesn't it sound so cool? And I'm like, yeah, actually it does. So she's amazing in her own way, and she was always there to kind of push me, stop me from doubting myself too much, because always
Mm-hmm.
as a woman, again, like in these, especially I guess in engineering, obviously there's not too many of us in engineering, and you always get these doubts. So she was always there to push me through them and give me the confidence that I needed to, just in general, just through my degree and through like this work now. And she's like, yeah, like she's always been there to kind of support me, which is, yeah. And I, yeah, it's, she's, I don't know if she's like, if that is her being my role model, but I've always like admire her so much for what she does and what she's done for me, I guess. Yeah.
Big up Estelle's mom.
Yes. Yeah, she's amazing. Anyway.
What do you consider to be the biggest mistake in terms of your education slash like academic career, and how did you learn from it?
My biggest mistake in terms of
Mm-hmm.
my education.
Yeah.
Hmm.
Or maybe some like turning point that made you really like
Yeah.
maybe like changed your mindset or views you had on a certain thing.
Yeah. Okay, well, it wasn't so, yeah, I guess it wasn't so much a mistake, but yeah, it did lead me to kind of where I am now. And that, so again, when you do, this is why they're very important, but when you do sort of group projects or even just any type of sort of hands-on project in your engineering degree, you learn what you like and you learn what you don't like. And I guess, and I wouldn't consider it a mistake, but because I had a 4-year degree, and where I just, it's an integrated master's, and in my third year, I had a group project where that I just ended up, not hating, but I just ended up like losing passion for what we were doing, and I just didn't like the project in general. And that maybe could have been avoided if I had gone through, let's say, the because you have a project list at the beginning of the
Mm-hmm.
year, you kind of go through it, and it kind of, yeah, you essentially, I guess a mistake I did was, yeah, not going through the project list as much as I should have, and so ending up with a project that I didn't like.
Mm-hmm.
But in the end, that kind of led me to knowing, oh, I'm definitely, essentially the project was quite mechanical, yeah,
Mm-hmm.
and which is essentially, it made me realize, okay, I'm not going to do any kind of mechanical engineering, because you can do mechanical stuff in the medical field,
Yeah.
but I just ended up disliking it. And yeah, and then I kind of went into like more so medical devices, electrical slash software. So it did end up being kind of a turning point, and that, yeah, it made me realize I don't want to do this. I don't want to do mechanical engineering in anything. But also, I would say it's, in terms of mistakes, definitely. And in terms of, I guess this is more relevant to engineering degrees, definitely like really think about what you want to do as a project, because these projects are I cannot like stress this enough. They're like the most important part of your degree, because that's kind of where you learn the most, and that's kind of where you figure out, as I did,
Mm-hmm.
what you like and what you don't. So
Yeah.
definitely have a look at, let's say, this project list that you get at the beginning of the year, and try to think about it like a bit before you make a decision, which is what I didn't do in that year. Let's say I was like, yeah, sure, well, let's go with that.
Yeah.
Definitely think about it a bit before you actually decide. That's a mistake I'd say I did, and something that you should, yeah, avoid by looking into the projects and really thinking about them. So yeah, I'd say
I
that.
will definitely take that. I will definitely take that into
Yes, honestly, like, yeah, it's a good,
um
yeah, actually, I'd say like that's pretty good advice. Hey, well done.
hey.
Pat on the back.
What are the top three pieces of advice you would give to yourself, either in like high school, secondary school, or university?
So I don't know about your high school, but my high school like had nothing to do with software and just coding in general. Like really, yeah, there was nothing, there was no course, there was no kind of A level, there was nothing that allowed me to kind of explore software and like how to code and learn how to code and stuff like that. So looking back, I did struggle. I mean, obviously at the beginning of your engineering degree, you will learn how to code and stuff, but it would have been a lot more smooth sailing if I had some sort of experience before uni. So I would say if I had to talk to myself, my younger self, try to, kind of, yeah, again, my school didn't offer anything, but it would have been nice if I had looked online, because as I mentioned, there's actually quite a lot of resources, and if I had pushed myself to learn coding myself, but the thing is, yeah, I just didn't really know about it. So there's
Mm-hmm.
that. Number one, I'd say read more about STEM in general, because that's how this will be coding will be useful, because of course you'll be like, if you learn about different, let's say, products or projects, and you'll be asking yourself, oh, but how did they get there, how did they get here, and then you'll think of the skills needed to actually do this project, or do this research. So read more definitely about STEM, whether that be research papers or engineering. Yeah, you can have research papers in engineering. So research papers are a really good thing to read, but also they can be quite dense in terms of
Mm-hmm.
like, a lot of
Yeah.
information there. So maybe things like there was like New Scientist, these magazines that do go into science, but that don't really give you like all of the details. They give you a
Mm-hmm.
nice overview. So maybe like reading more about them through these publications would have helped a lot, because then I would have known, okay, actually software is actually a really cool thing, because you read more about it with these magazines and stuff. So number one, yeah, reads more about STEM in general through any platform you want, really. Number two, yeah, would have been nice if I had kind of pushed myself to learn how to code, especially
Mm-hmm.
as I'm doing it right now.
Yeah.
It would have helped a lot, I'd say, in terms of, as well, in terms of confidence at uni. Like, it's, if you didn't do anything beforehand, and you're like, can I really do this? And it's just like, yeah, it really messes with your confidence, I'd say, if you really like are struggling with coding, and just with anything. So I mean, but then everyone isn't on the same boat. But anyway, but going back to the question, so yeah, pushing myself to learn how to code. Well, first of all, read more about STEM, which would have then led me to maybe like figuring out, actually coding is really cool, and learning how to code myself. And also, like, number three, probably I would have pushed myself to get out of my comfort zone a bit more, because there were quite a few, I guess, clubs that I could join, such as in my school, the debate club, or, and I don't know if there was a maths club. I don't know. There may have been something to do with software, some sort of coding club, and I'm not sure. Maybe there. I don't think so. But kind of pushing yourself out of your comfort zone will allow you to kind of explore just life in general a lot
Mm-hmm.
more. And let's say if I had joined the debate club, I probably would have been a lot more comfortable in presenting things, because obviously, if you, at least if you're one of the speakers when you debate, and it's like you talk in front of everyone. So I'd say, even if it doesn't seem relevant to you now, like, what would I have done debating something, some question? It's actually like, it just developing your skills, like just your general sort of skills, is actually a really good thing. So kind of, yeah, if you're unsure about joining that club, because I don't know, they do some random things, and you're not sure you would like that, just go for it. I'd say, like, at least when you're still young, you have time for these things as well. So yeah, I guess as the summary, read more about STEM in general. This is more specific to me, because I do software now, and software is lacking, I think, it's still lacking in schools. Push yourself to learn how to code. It's going to be a very useful skill in anything you do, honestly. Three, get out of your comfort zone, and join these, try to get these experience. Like, yeah, it doesn't have to be club, or it can, at your school, it could be something else, outside of school. Definitely push yourself to kind of do something you wouldn't really do. I mean, if you're interested in it and you're unsure about it, that's when I would have been like, go Estelle, go do it.
Do you think at uni you had pushed yourself or put
Yeah, I'd
yourself
say
in?
I pushed myself a bit more, but uni is also very stressful. So there is, I guess, let's say, a threshold to how many things you can do at once. Yeah, of course, there's your main degree, but then I also got involved in, of course, football in the Lebanese society, because I'm Lebanese. That's about it, to be honest. Those are the main two clubs that I was involved in, but to be honest, it seemed enough at the time. Another thing I would recommend is, try to get elected into, one of the committees of let's say your favorite club, because I was like events officer for the Lebanese Society, and that actually like helps you out with your organizational skills as well, because you're juggling that and your degree, and you'll just learn quite a few skills with just managing your time. Another thing I did to kind of push myself was I applied to this hackathon with my friends. It was like quite stressful actually, but essentially it's just like you trying to create a product or any sort of product in like 48 hours, right? That's kind of the concept of a hackathon. And that kind of was like, that was a really cool experience. And I'm glad I pushed myself to do that for sure. So it definitely got better, definitely as at uni, but it's something I definitely lacked when I was a bit younger. I just kind of focused on school and then hung out with friends, it wasn't, and I did some sports, but I didn't push myself as much as I did at uni. And even now actually, I did get a bit more lazy in terms of pushing myself when I started working. So thanks for like making me realize that. Cool.
How would you define success?
Okay,
Great question.
like again, going back to my mom, she told me this. She always tells me this actually. Like success is essentially like she's like doing something you love. Like she's always said that to me, and it's like stuck to my mind. And it's like, and essentially that's kind of how I define success. If you're happy, if you love what you're doing in terms of work, then you will end up being successful in your own way, because you will, if your passion is in what you're doing right now, then you may not, I don't know how you look at success, but essentially you'll end up in a place where in, let's say, 10, 20 years time, you're going to be super happy with what you're doing still. And you're going to kind of be successful in that way. Do what you love, and kind of the success will come later. Just in what it will lead to in terms of, of course you'll get, if you love what you do and you're doing well, you'll get that promotion, you'll get more money, whatever however you define success, like in terms of material things, that will come as a side effect of doing the job you love.
Yeah.
Yeah.
Really.
That's definitely like my mom again drilled that into me, and I agree with it. So that's how I
Yeah.
would define success, let's say.
So that's all the questions I have for you today. Thank you
Okay.
so much for joining me.
It was a pleasure. Thank you for having me, and I hope any person watching and I've interested them and I've helped them in some way.